Back to Europe in 2018, he worked in Barcelona and in Cambridge, where he conducted large-scale comparative analysis on birds. Simon finally moved to French Polynesia in 2021 as a researcher for the French Institute for Sustainable Development. There, he is developing a research program focused on invasive species in an eco-evolutionary framework. The role of behavior in shaping animal responses to environmental changes has been central to his research for over 15 years. Today, we welcome him for a presentation titled Adapting to a Changing World, Behavior at the Forefront in Birds and Terrestrial Crabs. Welcome, Simon. Thank you for joining us today and sharing your research work.
Well, thank you very much. And yeah, first of all, thank you to the organizers for giving me this opportunity to present my work here. So I'm going to present you some work I've been conducting on the importance of behavior and how animals respond to the current environmental changes that are due to human activities mostly. And so I'll organize that talk in two parts. One will be presenting results that are published now of large-scale comparative analysis. So that's going to be for a short part of the talk, but most of my talk is going to focus on work that I've been conducting here over the last 3 years in French Polynesia, and that's really focused on one case study where we are taking profit of a atoll scale, so eradication program. So really we have in situ experimental approach to understand how removing invasive species affect the functioning of an ecosystem, and especially how it affects the behavior of seabirds and terrestrial crabs. So the importance of behavior in shaping animal response to changes has been investigated for quite a while, and is acknowledged as potentially being one important part of these toolbox that animals have to respond to changes in their environments. And during my research over the last 15 years or so, I've been focused on different questions related to that general thematic. And today I'm going to tell you about two of them, especially the first one here that you can see on the screen, aimed at understanding how behavioral plasticity may improve a species' ability to respond to these changes at a global scale, so using comparative analysis at the scale of an entire class, the birds, basically. And in the second part of my talk, I'm going to talk more about how clearly animals use their behavior to respond to changes. How do they change the behavior in response to changes? Something many of you are probably more familiar with. Finally, what really is orienting my research recently is how these changes in behavior may affect the functioning of ecosystems. So not just how behavior changes in itself, but how this has consequences at the scale of the entire ecosystem. But that's something I'm not going to have much time to talk about today. It's more the general context here. But the first time of these talk that I was mentioning is going to focus on these large-scale comparative analysis, aiming at understanding the role of behavioral plasticity in animals' response to changes. And in the second part, we'll focus on a case study. So I'll bring you here to French Polynesia to look at how removing invasive rats from an atoll impact the behavior of animals, how animals, both seabirds and crabs, are able to respond to this removal of the key species from their habitats. So let's go for the first part of it. So it's been well known for a little while now that there is big discrepancies, big variation between species in terms of how sensitive they are to changes in their environment. Some species are at very high risk of extinction, others are doing relatively well today, and others are even doing very well facing changes. We've known for a while, including by a few studies that I've conducted with my collaborators, that things like ecology, so the habitat breadth, like life history, how fast you reproduce, for example, and intraspecific variation, so how many different strategies are existing within your population, will affect how likely you are to do well under current environmental changes. What is much less well known is how behavior matters, how important especially is behavioral plasticity. That's something that is expected in theory, but for which we have very little evidence, at least at a global scale. So why should we expect behavioral plasticity to matter and to help species to deal with changes in their environment? So to respond to that question, there is one context, one theoretical background that's really interesting, which is this cognitive buffer hypothesis, and which expects behavior to allow you to increase your survival, behavioral plasticity, to help you to increase your survival when facing changes. And that can be because you're better able to take profit of new opportunities as new food type arriving in your environment. It could be because you're better at switching to a new resource if your previous resource is not available anymore. Or it could also be, for example, if you're good at adapting your behavior to the arrival of a new predator or of new competitors. And these cognitive buffer hypotheses that has been developed since the late 90s suggest that this plasticity, so this ability to change your behavior in response to these new conditions in the environment, to changes in your environment, will help you maintaining a high survival, even in new and unusual environmental conditions that you haven't been exposed to in your evolutionary history. So that's the idea that's really behind this concept. Surprisingly so, today, if we're looking at some of the most plastic species, and I'm thinking about primates like these chimpanzees or these black macaques from Sulawesi that are very plastic in their behavior, able to really design new behavior frequently in their environment using tools and so on. These species are really doing very poorly today in the current environmental changes. We have similar examples in birds as well. These very charismatic Kia parrots from New Zealand are very good at using tools and have some extraordinary behavior as well that have been demonstrated. But we are also seeing them going very poorly, doing very poorly in front of current environmental changes. All these species are at pretty high risk of extinction. So that really raises the question of whether behavioral plasticity is actually helpful at all. And to respond to that question at a global scale, one key question is how do we do to measure that in a way that's comparable across species and across a large number of species if we really want to draw general patterns here. To respond to that question, I've been developing this work in the lab of Louis Lefebvre and then collaborating with Daniel Sol. We just used that idea that Louis had, so a professor at the University of McGill in Montreal, that you could just use the occurrence of the frequency of observations of behavioral innovation in the wild that are reported in the literature. So how do you define here behavioral innovation? You focus on two types of innovation that are basically bringing new food and new foraging strategy in a species repertoire. So it can be new food types in the diet, can be the use of new searching and handling technique. And Louis has realized that bird watchers and ornithologists in general are very keen on reporting these new behaviors in the literature. When they see a bird doing something unusual, they will very easily publish that because it's something that's unusual and interesting. So we end up having information on these behavior from the literature. This is just to give you one example of such an innovation. So these bullfinches in the Caribbean have been observed opening sugar packets. So they're just coming on these coffee terraces, and they are associating these sugar packets to the presence of food, and they also are good at opening them. So clearly this is a new food source that arrived there as humans brought these sugar packets on the islands, and they were good at taking profit of these new opportunities. So Louis realized that in all these journals of ornithology that you find all across the world, some very localized, local field ornithology journals, you can find lots of examples of these original behavior that have been observed only occasionally and are potentially new to the repertoire of these species. So in the analysis that we conducted next, we used these 3,800 examples of foraging innovation that he extracted from over 200 ornithology journals. But note that in between, Louis has updated his database, and now this database is published and publicly available in that paper published in 2020, where there's 216 journals that he went through, covering more than 60 years of ornithology and over 4,400 innovations. So a very, very large database, basically sensing all the examples of behavioral innovation related to food, feeding innovation in birds. So we used this previous database before the final update to look at how innovativeness uses a cue for behavioral plasticity, the ability to change your behavior in response to changes in your environment, potentially. How does that relate to global indices of sensitivity to current environmental changes? So as these were available by the IUCN, we really use these indices of extinction risk, and of population trend at a global scale that allowed us to work at the scale of an entire class. So we had more than 8,000 species of birds for which we had all the trait data that we needed, and on which we could look at how innovation rate and invasiveness, so whether a species was observed innovating or not in the wild, in the literature, whether that predicted their risk of extinction and their population trend. And we found that yes, indeed, the species that are more innovative are more likely to be doing all right and not be at risk of extinction. And similarly, they were more likely to have populations on the increase. This has been a very long work. It took almost 8 years to get there because we had to develop this index of innovativeness and innovation rate understand what it was related to, what kind of biases were behind it, including things like research efforts, and also understand how to evaluate other threats that may be related to this index of innovation, including things like habitat breadth, diet breadth, and so on. But the final result is here and really shows that innovativeness as an index of behavioral plasticity is one of the key threats that's predicting the risk of extinction in birds, together with things like generation length or habitat breadth. And interestingly, we found that these innovativeness and rate of innovation were good predictor of how species sensitivity to habitat destruction specifically. So it's not really buffering the birds against any type of threat, but really against habitat destruction. It's not really affecting their sensitivity to overexploitation, so mostly hunting and trade, or to invasive species, but really help them dealing with the destruction of the habitat. If species are exposed to habitat destruction, being innovative is helpful and help them decreasing their risk of extinction. So with this global scale comparative analysis, we really were able to bring an answer to that question of how important behavior is and really add behavioral plasticity to the list of threats that really matter in determining why some species are more at risk of extinction than others. And that's the end of this first part. I'll move on to the second part of my talk, which will focus on an in situ specific example. So in French Polynesia, on the atoll of Tetiaroa. So after working on this global comparative analysis, one thing I realized was how one threat specifically, the introduction of invasive species, has had a massive impact on recent extinction. They've been the main driver of extinction on islands, and islands are the areas where most extinctions have occurred, especially for groups of taxa like birds, for example. What we know is that both the invaders and the species that see their habitat invaded by newcomers are altering their behavior in response to arriving in a new environment or to the arrival of these alien species. There is this review paper that you can see here that shows on the right side of this figure the number of studies on invasive species on invasive population of the different taxa that altered the behavior or that investigated change in behavior. And on the left, you can see similar changes in the native species of the same taxa. What is really interesting is, first of all, that you will see that there's a lot of evidence for these changes, but what we are really missing is in situ experimental evidence. Lots of these studies are looking at how populations with and without the invader are different, or invasive and native populations are different. There's way fewer temporal follow-up of how populations are changing around the arrival or the removal of an invader. And this is where it is really interesting to work around these eradication programs. So rodent eradication is becoming one of the key tools to preserve biodiversity, especially on islands, for two reasons. Rodents have massive impacts, so removing them is really resulting in big gains in biodiversity, big conservation gains. And second, we know how to do it. It works when we are able to remove rodents from islands, especially from small islands. The two good reasons to do it, basically, today. So there's lots of programs that have been conducting these eradications, and positive effects have been demonstrated in many cases. However, we still lack a good mechanistic understanding of how the ecosystems are responding to these eradication, and yet they are some kind of ideal experimental setup where you're just removing the invasive species, and you can look at how the animals are responding to it. You can look at how the ecosystem is responding to it. And that gives an opportunity to understand how important behavior is in this kind of context. What happens, how animals change their behavior in response to the removal of these rats. So when I arrived in French Polynesia in 2021, I heard of a rat eradication program that was happening just 50 kilometers north of Tahiti. So I'm based there, as you can see on the top right, in the middle of the Pacific Ocean, and about 50 kilometers south of Tetiaroa is the island of Tahiti. So in Tetiaroa Atoll, we have this really interesting setup where there are 12 different islets. We call them motu in Polynesian. And only one of these islets is inhabited. All the others are conservation lands, basically. And interestingly, they have a relatively diverse history of invasion by rats. So we have two islets that were invaded by Rattus excellens, which is a Polynesian rat, a small rat species that were brought in by the Polynesians, the first human settlers, and that are relatively small, less arboreal than other rat species. So they're not as impactful as the bigger rats, and they're not necessarily going as much at the top of the trees to get bird eggs, for example. So you tend to still find a few birds on the island that are invaded by that one species. Then we have a set of other islets that had two rat species of the same Polynesian rat, but also the black rats, also called the sheep rat, that was brought in by the Europeans, with the first European explorers in the 18th century, and probably arrived on the atoll a few decades ago, so much more recent introduction. But these species is way more impactful. They're bigger, they're more arboreal, so they will go up to the very top of the trees to get eggs and chicks, and so potentially have a higher impact, especially on the fauna. And interestingly, we had some islands with only one of these two species, some islands with the two of them, and we also have three very nice control islands that have never been colonized by rats, which give us the opportunity to look at what's going on these islands when we're removing rats from other islands and have a control, so that we can really tell what we observe around eradication is not due to just a temporal pattern that's happening everywhere, independently of that eradication. So when we arrived in 2021, the rats had been eradicated from 3 islands already, but we were 1 year before the eradication of the remaining invaded motu, the remaining invaded islets. So we had a chance to collect data for 1 year and look at what was going on around the eradication. And we're still collecting data now to keep documenting the response of these systems to the removal of rats. One thing they haven't mentioned is that we're here in the middle of the Pacific, in an area that's so far from the continents that there's never been mammals that arrived naturally to French Polynesia. There's not even a bat species here. So all the mammals that arrive here were introduced by humans, and most of the function of these ecosystems are taken care of by other animals, other organisms, and that includes things like seabirds and terrestrial crabs. We'll talk about them in a minute. So importantly, around this global eradication program that's being conducted by a local NGO we're working with a lot, the Tetra Society, so that's in charge of managing this atoll. The objective is not just to remove rats, it's also to understand the consequences of that rat removal at the scale of the entire atoll. So the idea is that if you remove the rats, you will allow the birds, the seabirds, to come back, and the seabirds, because they are feeding offshore, sometimes several hundred kilometers away from the island, they then come back to the island and they concentrate a lot of the nutrients. So all the fish and squid that they're fishing end up via the bird poop, so via the guano, fertilizing these ecosystems. And it's a really important fertilizer in the ecosystem of an atoll, which is mostly dead coral. So coral skeleton, very limited in nutriment, very nutriment poor. So if you remove the seabirds, you're removing the fertilization of that system. So by removing rats, we're imagining that we'll add more seabirds, bring more nutrients here, fertilize the system, and increase the dynamic of that ecosystem. Will increase the dynamic of the vegetation, but also the flow of nutrients from the terrestrial area to the lagoon around and up to the coral. So really not just the terrestrial, but also the marine area. And so these organizations, this NGO we're working with, has gathered research groups from all over the world that are working on all the different compartments of this atoll, including in the terrestrial environment. And we're really working in collaboration with all these different groups, especially with groups that are interested in what happens in the terrestrial environment and how that can affect and influence what's going on in the lagoon. So our project is mostly focused on 2 faunal groups. So the seabirds, you can see here on the top, and the terrestrial crabs. So the purpose of our project is to understand how these 2 groups are going to change in response to the rat presence and the rat removal, and also not just how populations are changing, which is something that's usually documented, especially by conservation biologists, but also to understand the mechanism by which they're changing. Are they changing their behavior as well, especially, and how important is behavior in these response to the removal of rats? So we're investigating that on the seabirds and also on the terrestrial crabs. And ultimately, the goal of these projects is to understand the consequences of these changes in seabirds and crabs on the functioning of the ecosystems, on things like the nitrogen flow, we just talked about the seabirds guano, but also things like litter decomposition, scavenging, dispersal, and so on. I'm not going to talk so much about that, but I wanted to just make it clear that this is the purpose of it here, understand how changes in these 2 groups will affect the functioning of these ecosystems. So as we go to seabirds, I'm just going to show you a few key results, but we'll move very quickly towards the terrestrial crabs. We've been conducting monitoring for 4 years now. We are for a bit more than 3 years. We're in the 4th year of continuous monitoring, 1 year pre-eradication and 2.5 years after that eradication. So we're monitoring all of the 12 islets. we have 35 kilometers of transect that we're doing every 3 months to count exactly how many individuals there are around each islet. That allowed us, before the eradication, to show that the density of seabirds was very different according to the invasion history. There's way more seabirds in areas that have no rats and no ants either. I haven't talked about the ants here, but ants are pretty much the same issue. We have a yellow crazy ant invasion on the atoll that's almost under control now, but that's used to be a big issue for seabirds as well. And clearly there's way more seabirds in areas that have neither rats or ants. But in addition to just knowing whether the number of birds was different according to the invasion history, we're really interested in knowing how these birds are changing the behavior and especially the nest size selection in response to this invasion history. For that, we focus on 2 species, the brown noddy, which is one of the most common species on the atoll, so a very important one in terms of nitrogen input and phosphorus input. And we found on that left figure here that the density of nest per square meter was very similar to the global variation in nest densities. There's way more nests in the control area than in the areas that are invaded. If we're looking now at the nest height, so where do the birds nest? We found that they were much more likely to nest high up in the trees the moment there were rats present on these islands. And so the reason for that is that potentially when they are way higher up and the trees are more likely to have surviving eggs and chicks that won't be reached by the rats. So I forgot to mention that this is a really interesting species, these brown noddies, because most of them tend to nest on the ground when there's no rats, but the moment you have rats around, they are only nesting in the trees. And if you have too many rats, including black rats, we tend to just not find them at all, as you can see here on our last point with black and Polynesian rat presence. So really an impact on where they decide to nest. And this is data from November 2021, so that was before the eradication. But if we look at this information a year later, I'm not showing you the data here, I don't have the data out yet, but we observe a very similar pattern. It's just slowly changing towards individuals coming back to areas where they used to be rats. We then looked at that other species, the white tern, which is a pretty interesting seabird that's laying eggs directly on the branch without building any nests. And again, the chick here has very long claws that helps him maintaining and holding to the branch to avoid falling down. We've been monitoring that species as well it's a 4th year now. And the first result we obtained that was pretty striking was that just between the year before and the year after the rat eradication of 2021, we observed the doubling of the number, sorry, the eradication in 2022, a doubling of the number of breeding pairs on the islets where rats were eradicated, even though that number was stable in our control islands. So there's been a substantial increase in the number of breeding pairs, and it's not necessarily an increase in the actual population, but it's probably an increase in the survival of these chicks and eggs, which we previously were not seeing at all because they had already been predated when we were walking around counting birds. So a substantial increase in survival and probably in breeding success. Interestingly, 1 year post eradication, we also found that these species, through something that Antoine, who's now doing his PhD in our lab, detected a difference in the nest height as well in that species. So they always nest on branches, but they can nest very high in the trees or pretty low as well, sometimes just at 1 meter high or so. And in areas where there used to be rats, they keep nesting pretty high, around 9 to 10 meters high in the trees, whereas in areas that have never had rats, the average nest height was around 3 meters. So a big difference in nest height, probably as well as a way to avoid predation by rats. And you can see that there is here a legacy effect of rats, because that's 1 year after the eradication. And that's not so surprising because we know that this species is very faithful to its nesting site. We will come back to lay its egg at the exact same spot as it did the previous year. That's it for the seabirds. So we clearly see differences in behavior that are probably good ways to respond to the presence of these rats, but also will have effects on how quickly they can respond to their removal. So let's move on to these terrestrial crabs. And so first of all, why are we so interested in terrestrial crabs? That might be a bit surprising if you're looking at this general review paper that really is looking at all populations that have been monitored after rat eradication of different animal species, different taxa, etc. Most of the work was conducted on birds. There's very little work on invertebrates. And there's only 2 populations of land crabs that are included in these large-scale analyses, that meta-analysis. So that's a bit surprising. Why focus on land crab then? Well, clearly because land crabs in these remote tropical atolls are a key faunal group, very important for many reasons. So when you're getting on one of these atolls in the middle of the Pacific, that I was mentioning earlier, there's never been mammals there except for these introduced mammals, but you have very dense populations of terrestrial crabs. Very dense, very diverse. Some species are only in some types of habitat, others are more generalist, etc. So we have a very interesting community that is very important visually. You can see them everywhere, and sometimes in very large groups, that you can see here. They also are in charge of many ecosystem functions. They're the ones who will aerate the soil by digging burrows, disperse and predate the seeds, main scavengers, help in the litter decomposition, and they also are key predators. They will predate each other. So one between crab species, but also within crab species, there's a lot of cannibalism. And they also are predators of things like fish and seabird eggs and chicks. You can see here on the top right image, a caveo, a coconut crab, that's about to eat and predate a brown nutty egg. That really underlines how important these crabs are in these systems, and yet we don't know much about how impacted they are by land crab. There's a few studies that are showing very specific impacts, measuring one parameter and in one species that allow us to say, yes, there is an impact of rats on these crabs, but we don't have a very good understanding, a very integrated understanding of these issues. And especially, we don't know how important behavior is in these crabs. How are they able to change the behavior in response to the presence of these rats, for example? And these could have consequences also to interpret crab monitoring data, because the way you monitor land crab in most cases, you just go and count crabs along transects in some areas at a fixed time of the day, and you do that in areas with or without rats, or before and after removing the rats. And the number of crabs you observe is considered as indicative of how many crabs there are in the population. And if your rats are affecting the activity of your crabs and how likely they are to be out and active when you're counting them, this is clearly a bias in a biased interpretation of your data. So we need to know better how this behavior is altered by these rats, among other things. So one key part of our project here aimed at really documenting the changes in the land crab communities at also the population and individual level. So we measured a set of different parameters in order also to characterize changes in behavior. One thing to keep in mind before I go any further, that also they are mostly terrestrial. These land crabs all have a marine phase. So at some point, the females would lay her eggs and release larvae in the ocean that will mix in the open ocean between population of crab that come from rat-invaded, rat-free islands. So it's really unlikely that you get any local adaptation. If we detect differences between rat-free and rat-invaded area, it's more likely to be some filtering effect, intrageneration selection, or a plastic response to the presence of rats, which could be a behavioral plasticity as well. So on Tetiaroa, we've detected and identified 12 land crab species since 2021. Some were only present in areas that were rat-free, others were pretty much everywhere. So we are monitoring all of these species, but we had a special focus on two groups of species that were especially interesting because they were present on both rat-free and rat-invaded area. If we wanted to look at how they change after the rat removal, we wanted to have some species that were already present in areas with rats to really understand how rats were impacting them in terms of threats, including behavior. The first group of species were ghost crabs. We mostly focused on one species of ghost crab because they were the most abundant, which is a sedentary species specialized in these sand beach area. They tend to be sedentary at defending their burrows. They're also quite short-lived, living about 3 years, and it takes them about 7 to 8 months before the moment is settled, so get out of the water to become of a decent size and potentially reproductive individuals. We expect them to be preyed upon by rats also. That's something that we want to demonstrate here. The other groups were hermit crabs, who have multiple species here, including this strawberry hermit crab that you can see here, this red guy. That's a group of species that are carrying shells, so shells of mollusk. And they, therefore, are basically walking around with their shelter on their back. So they're way more nomadic, not territorial, habitat generalists as well for most of these species. And they're long-lived. They can live several decades, which is very different from the ghost crab. And we expect them to be more in competition for food with the rats based on data from the literature, although there might be some predation happening as well. One really interesting trait for the hermit crabs here too, in terms of behavior, is that when you put them when you scare a crab, when you disturb it, it will hide in its shell, and you can then measure how long it takes for them to get back out. So it's pretty easy to measure the boldness both in the field and in the lab by just putting them on their back and look at how long it takes for them to get back out. So let's start with the ghost crabs. So with the ghost crab, we really wanted to use that as a model species here to characterize the effect of rats on the risk of predation, on their abundance, on their sex ratio, morphology, and on their behavior by measuring their pattern of activity and look at whether they avoided being active when the rats were around. We had two levels of comparison, measure differences between islets with and without rats before the eradication, and then look at how it changed before and after the eradication. So these are preliminary results I'm going to show you. There's a lot of these data that's not entered yet, a lot of the data that we're still collecting. So most of the results are going to focus on the spatial difference, and there's going to be a few information on the temporal change after the eradication. So we measured the risk of predation by tethering crabs in the field. We measured the population size, sex ratio, and morphology using capture mark recapture approaches. And we also measured the daily activity patterns. So when are they active? Are they more active at some moment of the day than others? And the relative activity. Are they more active once we've removed the rats than they were before? So let's start with the predation risk. So how did we measure that? We collected crabs all from the same beach. We tethered them by just gluing a fishing line of a meter that's a meter long. And then we set them up on beaches all around the atoll. And they are within the ghost crab habitat, so they can live their ghost crab lives here, big burrows, etc. And we come back 24 hours later to see whether they survived the 24 hours period. We did that just before the eradication, and again 7 to 8 months after the eradication to understand how they changed in response to the removal of these rats. So before the eradication, we found that the survival was very high for crabs that were on rat-free islands. So the 2 points you see on the left here, we have a survival that's around 90%. And on the right side here, the black dot in areas that were invaded by both black and Polynesian rats, we see a survival that's closer to 20%, so much lower. And the areas, the two islets where there was only Polynesian rats are here intermediate. So very likely we have an impact of rats on their survival. We also had cameras on some of these individuals so we could tell who was eating them, and we could confirm that indeed rats are actively eating these tethered crabs. Interestingly, after the 2022 eradication, we're talking about just a few months later, 7 to 8 months later, we didn't detect any change in the survival of our tethered crabs in areas that did not have rats before. So the two black points on the left of this right panel, but the other 2 points on the right here, we had a massive increase in survival, clearly related to the removal of rats. So we're just removing a main predator on these individuals. So that's really good evidence that these crabs were predated by rats. So here we're working with tethered crabs. So it's not exactly the same as the general rate of predation, but this is in line with observation we had in the field of rats actively chasing and eating ghost crabs, including by digging them out of their burrows on beaches at night. So these very high predation rate really made us wonder whether the rats were having an impact on the size of the populations and the density of ghost crabs. So to better understand that, we ran a set of capture mark recapture sessions in areas with and without rats, and we did that again a few months after the eradication for a subset of sites. So here again, this is just very preliminary results. But the first thing that we noticed was that in areas that had rats, we were capturing fewer crabs for the same capture effort, for the same effort of crab search, basically about twice as many on rat-free islands as compared to rat invaded. We're now looking at the results taken out of mark. Before the eradication, we did notice the areas that had the two rat species tend to have a lower density of ghost crab, whereas areas that had never been invaded, especially, had a higher density of crabs. Now, if we're looking on the 5 sites on which we did these CMR sessions again, so 7 to 8 months after the eradication, we could tell that there were no change in the two sites that you can see on the right. So here we're comparing in black the same site before the eradication with the site after the eradication in gray. We can see that these Rattus excellens eradicated 2020 and the never invaded area, there's no significant change in the population size of ghost crab. But if I'm zooming in now on the 3 sites on the left, which is what that figure is, just to zoom in, we can see that there's a doubling of the ghost crab population in these areas where we removed the rats. So removing rats really allowed this population to increase substantially and that's just 7 to 8 months after eradication. So in parallel, these capture mark recapture sessions were a good opportunity to get some morphology data. So we could notice, here I only have data before the eradication. We could notice important differences in the size of the crab. The crab were much smaller in areas where rats were present, in areas with no rats. And they also had a significantly bigger claw. Here we're talking about the relative size of the claw. So we're correcting that size by the length of the carapace. So for a given carapace size, crabs would have a larger claw if they were on rat-invaded area, like if they were using these larger claw to defend themselves against rats. So there is some selective pressure happening here. It can either be a filtering effect where individuals with larger claws are selected there within a generation, or it could also be more of a plastic response to the presence of rats, something we don't know at this stage. Very quickly here, in parallel, we didn't find any difference in sex ratio, which is something that could have explained potentially the difference in claw size, because males have bigger claws than females, but that's not something we detected between rat absent and rat present areas. In contrast, we found a difference in body conditions. The relative mass of our individuals, when we correct for their body size, they were in a better body condition in areas with no rats, which suggests that potentially they don't have as much time to look for food and be active in areas where that were rat invaded. So we decided to also try to measure variation in activity in our crabs. So we didn't just try it, we did measure it. So how did we do that? We set up cameras, trail cameras all around the atoll in areas with and without rats before and after the eradication. And with these cameras, we were just taking one picture every 15 minutes of an area of known size. So on the first picture, we set up these 5 by 2 meter quadrat that allow us to then look at each picture, count how many crabs are active in these 10 square meter. And we can do that every 15 minutes, which allows us to look at when they're active during the day as well. And we did that just before the eradication, just a few weeks after the eradication, to look at immediate changes in behavior. And then again, 7 months after the eradication. We keep doing it today again as well, but these are the data I have available now and I can show you. I'm going to pass on these methodological slides to right away go into the results. And this is information on the relative activity. So how many crabs are we seeing active on our photos? These two panels are basically showing the same result using different metrics of activity. So if you look at the left two dots, you can, sorry, if you look at the gray dots, you can look at change in the number of active crabs in areas that were invaded by rats. You can see that there is an increase between just before and just after the eradication. Here we're talking about 4 weeks of difference between the two measurements. In contrast, in these black dots, so areas that were rat-free, where we did not remove rats, we looked at their activity at the same period and we did not detect any significant change. So removing the rats really increased the number of active crabs here, which is an important result, I believe, to understand how rats are affecting crab activity. More interestingly, to my opinion, we also looked at how and at when the crabs are active during the day. So to that aim, we looked at the daily activity pattern. So let's focus on this left panel, which is a pattern of activity in the rats. It goes from noon one day to noon the next day, with midnight in the middle. And you can see that the rats are mostly active at night, with a peak of activity just after sunset and another one just before sunrise. We're now looking at the average activity pattern of a ghost crab. We see that they also are mostly nocturnal, but they have similar peaks of activity around sunset or sunrise as the rats have, which might be an issue for these ghost crabs that are predated by rats. So now what we did is that we just split our crabs into two groups, a crab that were occurring on rat-invaded area and the one from rat-free area. And we found that the two types of crabs had very different patterns of activity, noticeably because crabs from rat-invaded area tended to avoid being active when the rats are the most active. So interestingly, we could keep monitoring this activity across time. So immediately after the eradication, and then again 7 to 8 months after the eradication. And that gave us the opportunity to look at how the crabs from previously rat-invaded area changed their behavior, changed their pattern of activity, and how it started to match better with an activity pattern of a rat-free area, of a crab from a rat-free area. And we found that the overlap between these two curves is slowly increasing across time. So we are removing slowly this difference in activity pattern after having removed the rats. But we're not quite there yet. 8 months after the eradication, they still show some difference. So just to quickly summarize his results on the ghost crab, we had a pretty good integrated understanding of how the ghost crabs were affected by rats and how they're responding to the removal of rats. We see that they are heavily predated by the rats. They also are changing their morphology in response to the rat presence, and hopefully we'll be able to document changes in morphology now that the rats were removed. We also show that there is a pretty good ability to recover already a few months after the eradication in these ghost crabs. And finally, they have these important changes in behavior. Some of them are immediate just after the eradication. So very clearly some plastic responses to the removal of rats. They're able to detect that there's no more rats around. And that raises questions in terms of the interpretation of some of the monitoring methodology. So that's it for the ghost crab part. Let's move on to hermit crabs. I'm going to go very quick on hermit crabs. So what we measured on hermit crabs was basically just compare their sleep all the time. So what's going on when we put them on their back, how quickly do they get back out of their shell and back on their feet. And we did that in the field, looked at individuals on rat-invaded and rat-free area, and we found that there was a massive difference. They're much faster at getting out of their shell when they were on islets that have no rats as compared to islets with rats. So we're talking about 10 seconds for individuals from rat-free area and around 40 to 45 seconds for islets with rats. Interestingly, when we measured that same trait 1.5 years after the rat eradication, we still detected this exact same difference. So the crab had not really understood yet that the rats were gone, or they haven't changed their behavior at least now that the rats were gone. We measured the exact same trait in captivity. So we just brought the crabs to the lab and looked at how quickly they flipped. Exact same protocol, but this time in more controlled condition. And we detected the same difference between individuals from rat-free and rat-invaded area. And finally, the last aspect that we measured on these crabs, this time only in the lab, still on the hermit crabs, was their exploratory behavior, exploratory tendency in these small arena. We're just looking at how quickly they start exploring the new arena and how many zones they are exploring in a given amount of time. And what we found was that this was predicted by the rat history. Individuals from rat-invaded area were much slower at exploring and took more time to start exploring. And interestingly, this had not changed either 1.5 years after the eradication. So there's a strong legacy effect. If you used to be on an island that had rats, you will behave differently than if you used to be on an island that did not have rats. So if you are on an island that used to have rats, you're not changing your behavior because the rats were removed. What matters is not whether the rats are present, but whether they were historically present or not. So just a take-home message for these crab results. So we detected very quickly that behavior can change after the eradication. Very quick changes in these ghost crab. However, these varies across species and may be related to life history. We've seen that in these hermit crabs, we didn't find a change in behavior even 1.5 years after the eradication. So potentially, because they're long life, they don't have the same ability to switch their behavior, but that's something that remains to be demonstrated and to be further investigated. Interestingly, why we're documenting all these changes that we imagine are related to the removal of rats, there's big changes in the structure of the crab communities and the population density. So this is something that will also likely affect how crabs are behaving. So these species you can see here, these fiddler crabs, used to be very hard to see, very uncommon on the atoll. And there is now a few areas where they're pretty dense. They probably were also predated heavily by rats. So these changes in the crab community will potentially have impacts on how the crab are behaving globally. I underline that some of our results, especially on behavior, may have consequences for the interpretation of these monitoring protocols. And in the future, what we're really hoping to keep monitoring is how behavior is changing in response to these crab community changes. We're noticing some species of crabs, especially these predatory crabs. You can see here the bottom left, Geograpsus, that are heavily predating on ghost crabs and that were not present in areas that were invaded by black rats. So now slowly we see a big increase in the population size of these individuals. They're getting more and more present, more and more abundance, and we are wondering how this is going to affect the behavior of our crabs. We're also wondering how this increase in the crab population and the crab density and crab diversity will affect seabird recovery capacity, because these crabs, we've seen it here, are important predators of seabird, especially chicks and eggs. And since some of the crab species are recovering very quickly, whereas the seabirds are much slower. We're wondering whether at some point the density of crabs is going to be such that new seabird colony will really struggle to settle there. But that's something we're investigating and following. And finally, what we really hope to be able to do is understand how not only the changes in these population and community structure in crabs will affect the functioning of the ecosystem, but also their changes in behavior. A crab that's more active probably has a more important impact on its environment than crabs that are just hiding continuously from rats. And before finishing here, I just want to put back here a more conceptual diagram, just that I just stole from the literature here, but just to put our work in this general context. We're really hoping to understand how behavior at the individual level, the change in behavior of an individual, but also the selection of some behavioral traits may have consequences in terms of how population respond to changes in the environment, and especially here to the presence and removal of invasive species. And the changes in behavior at the individual level can have consequences not just on the number of individuals, but also on the interaction with other species, the structure of communities, and ultimately on processes at the level of the ecosystem. And here in our case, even at the meta-ecosystem, when you're thinking about this flow of nutriments between the terrestrial and the marine environment on our lagoon, on our atoll. So with that, I'm going to thank you for your attention. I'd like to thank all of my collaborators, and especially Jana DeVore, who's been full part, 50% done 50% of the work for the whole study on this atoll that I've been describing here. And of course, I'd like to thank all of the people that have been helpful in the field, all of our students here, and the founding agency. And thank you again for giving me the opportunity to give the talk.
Well, that was a really interesting and fascinating experiment. And I guess this is the first time I'm hearing about a talk about crabs, especially. So that was really interesting to hear. So now people can post questions in the YouTube. So we already have a bunch of questions. Um, so Laura asked, very interesting. Do you know if the behavioral responses of birds facing rats was fast? So that means avoiding them and avoiding maybe breeding in those islands with rats. And if their behavioral response to the, I guess the question was not completed, but yeah. So it's like, do you know if the behavioral response of birds facing rats was fast?
Did they quickly change the behavior? Is the question, if I understand well.
Yes.
Yeah,
Yes.
that's an impressive question. We don't know that exactly because we're still documenting it at the moment. And after 3 years after the eradication, we still don't see a massive change in the seabird population yet. What we are seeing, one key question we're facing here in terms of their nest site selection is whether the same individual is able to alter the where it's nesting. Is it the same bird that 1 year will decide to nest on the palm trees, the next year will nest on the ground, or do we just have different individuals that are potentially imprinted on their own nest where they grew up, or on whichever nests they built in the past? So we don't know exactly if they're changing their behavior, or if it's just individuals with different behavior that are selected by the rats. So that's a good question. At this stage, what I can tell you that this is not changing very quickly, but that's something we kind of expected if we are in this scenario where the same individuals keep nesting in the same, having the same nest site.
Next question from Ellen. Thank you for the excellent presentation. In your study on innovation, were there differences in the type of innovation on success? Example, those related to food versus habitat versus predator avoidance?
Yeah, that's a good question. Yeah, I apologize if I wasn't very clear on that, but all the innovation we're considering here are food type innovation or food technique innovation. So different ways of foraging or different food items that are being incorporated in the diet. We only focus on that because this is the one that's the most frequently reported in the literature. So habitat related changes or changes in behavior, like potentially colonizing new habitats, etc., are way more difficult to get out of the literature, I believe. So that's why we're really focused on the food type. What we looked at is how different it was to be innovative in terms of just adding a new food item that was not known before. That can be, a granivorous bird that was observed adopting new types of seeds that were not in his regular diet before, versus an actual new foraging technique, like starting to eat sugar packets for a bird that used to eat nectar, for example, which clearly is a much more different technique of foraging. So we looked at whether individuals or species, sorry, that are more that are using more technical innovation were also more buffered against extinction as compared to species that are more using food type innovation. And it is the case. It looks like foraging technique or so which we would intuitively consider as more advanced behavior cognitively or more rewarding in terms of allowing these birds to do well in front of environmental changes.
Question by Anne Marie. Thanks for a great talk. Did the crabs aggregate more before rat eradication as a form of anti-predator behavior, such as safety in numbers or dilution effect?
Yeah, that's an interesting question. So my first answer would be no, they were not. But it's also very variable across species. So these ghost crab that I've talked about a lot, they are pretty small crab species as compared to other species we have on the atoll. And these ones probably have no advantage in aggregating. They're just going to be eaten no matter what. So it's pretty much what came out of our result. They're heavily predated. So the populations are much smaller when there's rats around. But there's other species that are more gregarious, that are living in big colonies where you just have dense burrow areas, especially the Scardisoma carnifex, which called the tupai here. And that species is probably a bit less sensitive to rats when they reach a given size. So you would see aggregation of individuals of the same size, basically big individual that survive the younger stage without being eaten, probably by hiding in the burrows of bigger individuals. What we are observing is not so much that they were more aggregated, but we observe differences in the size structure. We're seeing more and more small crab of these guys. We used to wonder why there were no small crabs of these big crabs. Where are the babies, basically? And we slowly see them hanging around. So before the eradication, they probably were either hiding or they were just being eaten. So there were way less recruitment into these populations. But because these crabs are long-lived and take a long time to develop, it's going to take us some time before we understand exactly how they react to what is a normal size structure in these populations. But not much difference in terms of aggregation. They're not very social overall, these crabs.
Question from Tang. How do birds and crabs differ in their adaptive behaviors when faced with similar environmental pressures?
How do birds and crabs differ in their behavioral response? That's a difficult question. I don't think that I have an answer to that. They are so different organisms. They are just focused on different lifestyle as well. They are they're very different. So it's hard to tell how they differ in their adaptive behavior, and at the same time they differ in pretty much everything. For the seabirds, they're foraging at sea. So when they're at sea foraging, they're not really impacted by rats. It's really when they're coming back to land to rest or to lay their eggs and breed that they are heavily impacted by rats. It's really a predation interaction that is happening there mostly on their young, on the eggs and the chicks. For the crabs, depending on the size of the species, they tend to be affected either by direct predation, including on the adults. That was the case in these ghost crab, but also potentially by a competition. We imagine, especially, we're trying to document that by measuring the isotopic signature of the different crab species and how it's going to change between before and after the eradication. So we're going to be able to have that information at some point on whether they change their diet, especially hermit crabs, once the rats were removed. So in that case, we imagine it's more of a competitive interaction. So they may change their foraging behavior, but we don't expect seabirds to change their foraging behavior because it's not something we expect to be impacted by rats.
So I'm interested to know why some of those islands were never impacted by rats or infected, I should say, or colonized by rats. So was there any reason that some islands were versus some islands were not?
Yeah, it's a very good question. The key reason we imagine, we don't know exactly when these rats arrived there. We don't even know if some of these introduction were intentional or not. It's probably likely that they were all unintentional. They just arrived on the boats of people who showed up there. We believe that what happened is that these islands were relatively small, so way less interesting for people to settle on them. So on these three islets, we don't have any good track of human occupation, for example, whereas it's the case for other islands. So the rats probably arrived in areas where humans arrived. I didn't have the time to, list you give you a history of the use of these islands. They've been used a lot by the Polynesian. Traditionally, they were a very important area of gathering of Polynesian from different areas, from different archipelagos. But they've also been used as a coconut plantation more recently in the early 20th century. So there's plenty of islets that have been fully cut. All the native vegetation was removed and planted into copra plantations, so big coconut trees all over. That's been abandoned since the 60s, so we can see native vegetation regrowing in these areas. But that also probably impacted where the rats are. And these islets that were smaller were not so interesting. They were also a bit more remote, a bit far away from the other islets, a bit more difficult to get to. That's probably explaining why the rats didn't make it there.
Also, did you try to understand what were the keystone species before the eradication and after the eradication, if it has changed in the community, or is it something you're planning to look at?
We're planning to look at which are the keystone species before
Yes,
or after.
yes, if it
Yeah.
changes due to eradication of a whole species.
Yeah, no, that's a good question. I mean, clearly we know that there's going to be a massive change, which is that probably one of the main keystone species were rats, and the rats are gone now. So that's also why it's so interesting to study that system. It's one of the rare opportunity to remove a keystone species artificially. Here, we just remove it and we look at what's going on next. So that's why it's so interesting too, and very when you remove a species from a from a community and ecosystem and say in a tropical forest in South America, you probably are very unlikely to detect any effect because the effects are going to be very subtle, very difficult to measure. Here, we're removing one species out of maybe 20, 30 species. The species richness is very low in these areas. So, and we are removing a species that we know has a very strong impact. So we clearly are measuring very quick responses at different scales, from the micro scale to more community and ecosystem scale. So clearly there is that difference. And we also are documenting slowly changes in which crab species are the most abundant. And obviously there is going to be a new balance that will be reached between these crab species that are interacting a lot, and potentially between the crabs and the seabirds. So yeah, we're hoping to document that, but it's going to take a long time, I think, before we get to a new equilibrium because of how long some of the species that we're hoping to restore are.
And as we are running out of time, one final question. So regarding in terms of application of the study, so is the French Polynesia now protecting or being more careful with invasive species, or how would you suggest people to handle invasive species in general, and how it impacts the community itself?
Yeah, I think it's in general, I think it's a study that aims at understanding whether the impact of removing these invasive species is just recovering seabird population or if it's having more broad scale impact. And I think our work is documenting at the moment that we're not just helping a couple of seabird population to recover, we are helping a whole ecosystem to become functional again, and potentially not just that terrestrial ecosystem, but also what's going on around it in the laguna as well. So that's a long-term study that we started here with this purpose, which is really to understand the consequences with a very integrative approach to understand the consequences at the scale of the entire atoll of just removing rats, basically. So that's something that's expected theoretically, and that has been shown using correlative comparisons. But here we have a chance of really pinpointing the mechanism using a causal relationship here. So I would say, yeah, that's something that hopefully will help convincing managers that removing invasive species, including especially rats, is having a massive impact. It won't just save a few seabirds, which already would be probably a good argument, but here it's more than just seabirds.
All right, thank you again, Dr. Simon, for giving your excellent presentation, and congratulations on starting your new lab. I'm pretty sure that people are going to have more questions, and if you have more questions, please post it on the Discord. I'm pretty sure Dr. Simon will be happy to answer them. For now, we'll be having a quick 10-minute break before we start with more presentations, so don't go anywhere. We'll be back after 10 minutes.